demonstrating how we're going to set up your bacterial lab that you're going to be doing. And so I've got my supplies out here. What I'm going to be testing are different types of disinfectants on the bacterium Serratia marcescens, or S. marcescens. And so the first thing I'm going to do is I'm going to label my Petri dish. This is a Petri dish. It's got a cover here and a bottom. The bottom has the agar on it. And so whenever you label, you flip it upside down, so you're labeling the bottom side. So I'm going to start by just putting an X here, like a cross, to divide it into the 4 quadrants that I want. Okay? And then I'm just going to label. So I'm going to test Serratia marcescens, because sometimes it likes those moist areas, so kind of like you might find it in your bathroom, in your shower or something. So I picked some things that maybe you'd clean your bathroom with, whether it's your showers or your floors or things like that. So I'm doing Clorox / Tilex shower cleaner. I'm just going to abbreviate that. Spick and span. Lysol. And then don't forget you need to have a control group. Okay, so I'm always going to leave that one up there like that. Now the other thing you'd want to put is the name of your bacteria you're using. So I'm just going to abbreviate it. Remember, S. marcescens, and I might even just do S. marc just to save a little space. But you know, obviously in your lab reports and things, you'd want to have that spelled out all the way. I'm going to put my initials. You'd want to put the date as well. So I'm going to put 9/24. Or was it today? 21st. Sorry. I think this Thursday when the first group goes, it's going to be the 24th. All right. Class period. That's good to put that down so that you know you can find your Petri dish, because we're going to have a lot of Petri dishes in there from different classes. So you're going to want to have it marked good. Once you've done that, really our next step is to get your bacteria onto your plate. So now I'm going to flip it over so that the cover's on top. I'm going to take a sterile swab. I'll swirl just a little bit there, S. marcescens, and I'm going to swirl my swab in there to kind of stir it up to make sure I get a good coating of bacteria. Tap it a little bit inside there before you pull it out so it's not totally dripping all over your lab station. While holding your cover at a slant to prevent other microbes from dropping in, now you're just putting this over the surface of the agar. It's like you're coloring it. Now I'm going to turn this slightly, and I'm going to do it again because there may have been some areas I couldn't reach as well with this swab. I'm going to turn it one more time, and I'm going to swab it again. So you get a nice coating over the whole plate. Okay? Once you're done with that, you're going to put it into a biohazard bag over here. And we'll have some of those dispersed throughout the room so that you have one close to your lab table that you can put that into, and then I sterilize that later to kill off the bacteria. Once you've got your bacteria on there, now it's time to put these different cleaners on. The way that we do that is we have these little filter paper discs that we punched out, and then we've sterilized them in the autoclave. And then we have these little forceps here, or tweezers. I have them soaking in some rubbing alcohol, so we'll have some rubbing alcohol at each of your stations. Before I pick up one of these discs, I take the tweezers out of my rubbing alcohol and I wipe it down really good. Okay, so it's nice and dry because I don't want any rubbing alcohol still on there, or else that's what will get onto your disc. I'll start with this floor cleaner. If you have something in a spray bottle, you can just spray a nice little, oops, off, turn it on first. You can spray a nice little pile right here on your lab table. It works really easy that way. Grab a disc, dip it into there, okay. Find the right spot here. I'm going to flip it back over, so now that's facing down. And now I'm going to put it right in the middle of that quadrant and tap, tap, tap to make sure it's secure. Don't dig into the agar, but tap it so it stays on there. Dip it into your rubbing alcohol. Dry it off good. Then I'm going to grab another, but first I'm going to put my spray out. Oops. If you lose it, then you just have to pick it up again. And let's see, spick and span is right in that quadrant. So there it is. Put it in the center of the quadrant. Tap, tap, tap. Rinse it off. Dry it off. I'm just going to pour a little bit of this. If something comes in the cap, you can just put a tiny bit of it in the cap, because like I said, you don't need very much of this. And dip that in there. And then the lice, oops. Where did I lose it?
It's in the cap.
This is stuck in the cap. I don't even see it in the cap. That's so funny. All right, I'm going to redo this. So see, things happen sometimes. I'll get a fresh one now. Try it again. Tap, tap, tap. Rinse it. Off. And then for my last one with the control group, remember the control group doesn't receive the experimental treatment, which means it doesn't receive any of these disinfectants. So I'm just going to grab a plain disc. I'm going to place that right into the middle of the quadrant that says control and tap, tap, tap. Now some groups are choosing to maybe soak that plain disc into some distilled water so that it still has liquid, but it doesn't have anything on it. That's perfectly fine as well. Whatever you and your lab partners decide that you would like to set up your experiment, then that's how you can do that for the control group. I guess I just leave that soaking in there till the next lab group comes in. Now we need to secure our Petri dishes, so we always put a little piece of tape on either side. This and rip it in half here. And I'm just going to put a little bit right here on the side and another chunk on the opposite on the other side. I always like to keep this area free because this is what you're going to look at to measure tomorrow. That's why the writing stays on the edge, the tape stays on the edge, okay? You're not going to open up this Petri dish again because we don't want to expose you to bacteria that has grown on there. So, but you want to be able to have a clear view of all of your discs and whether or not they have a zone of inhibition in it. So the last step then is to put this in the incubator. Serratia marcescens likes a little bit of a cooler temperature, so it's set at almost room temperature, 25 degrees Celsius. That's going to be this incubator right over here. We'll have it marked with both the name of the microbe and the temperature that we have it set at. You always place your dishes in with the bottom facing up because remember there's some condensation here on the cover. If you did it this way, that might drop down and ruin your experiment. So we always place your Petri dishes with the bottom facing up. And then, of course, as always, you would clean up your lab station before you leave. Wash your hands after this lab. Make sure your lab station is nice and neat and clean. And now you have to wait 24 hours to see how that bacteria is going to grow.